Bittu Kaveri Rajaraman is an associate professor of biology and psychology and is the current head of the psychology department at Ashoka University. He received a PhD from Harvard University in neuroscience and was then a DST-Kothari postdoctoral fellow at the Center for Ecological Sciences, Indian Institute of Science, and then an Inspire faculty fellow at the Central University of Hyderabad. He works on the evolution of neural and behavioral systems of communication, the neuroethology of temporal pattern recognition in insects, and quantitative and economic cognition more broadly in zebrafish, dogs, and humans. So take it away.
All right. Thank you very much. It's been a crazy couple of weeks. So apologies that I could really only send my sort of bio and headshot and so on sort of relatively late. And I know that and so I was able to commit somewhat late, but really glad to be here. So is this then 25 minutes and then 5 minutes for questions? Yeah.
Yeah. I'll tell you when it hits the 20-minute mark, and we can go ahead with that.
Okay, that sounds good. All right. Can everyone see my slides?
Yes.
All right. Wonderful.
It's not in presentation mode yet, though.
Ah, that's right. Yeah. Okay.
Perfect,
so
perfect.
What I'll be talking about today is the evolution of behavior and the underlying and the goal of our lab, which is to try and find the neural basis of behavior and to understand how these neural circuits sort of evolve in order to understand the mechanistic basis by which behavior evolves. Our approach is one where we try to integrate behavioral ecology, neuroscience, and neuroethology, neuroscience and behavior, and an understanding of the ways in which evolution is shaped by ecological pressures, and so on, right? So this is therefore a sort of integrative and sometimes slow and frustrating exercise. But I'll take you through what one of the systems that we work on, and I'm particularly interested in finding students who are interested in these questions and would want to work on these kinds of systems. All right. So the system that I work on is this system. This is one of the animals I work on. Now, if you live anywhere in India, you will have heard this animal at some point in your life. If you live in Sonipat, where I do, or in Delhi, you would have heard these animals. If you live in Bangalore or Hyderabad, or really anywhere in the south, anywhere where there's a decent amount of vegetation, you will have definitely heard these animals, this particular species, right? And so as you can kind of see, I'll just play that. If you look at the video, you'll see that, and it's a beautiful video taken by a student named Ronit at Ashoka University. You see this very clear kind of movement of the wings of these bush crickets, as they're called. That produces sound. There are two major groups of, acoustically communicating insects that fall within Orthoptera, within the larger group of Orthoptera. And those are locusts and crickets and sort of, locusts and grasshoppers, right? One group, and crickets and bush crickets, another group. Very often people confuse them. You see something that looks green is in the grass, and people think this is a grasshopper. In the old days, of course, actually crickets were called longhorned grasshoppers, and in that name lies one of the ways in which one can most quickly and informally tell whether the animal one's looking at is a grasshopper or a cricket / bush cricket. Grasshoppers and locusts tend to have these short, stubby antennae that you can see on the right. They're about the size of the face, right? Whereas if you're looking at crickets and bush crickets, they'll have very, very long antennae, sometimes, often as long as body length. That's one of the quick ways in which you can identify these. And of course, there are others specifically important to sound production, because we care about acoustic communication, is the fact that crickets and bush crickets, while sharing the same orthopteran lineage as grasshoppers and locusts, produce sound and hear sound in very different ways. Grasshoppers and locusts rub their back legs, right? And it produces a sort of kind of sound that you might have heard, on a hot day during the day. You sometimes hear these, this rustling sound from the grass. That's often a locust or a grasshopper calling. Whereas crickets and bush crickets produce sound by rubbing their wings together, as you might have seen in the previous video that I showed you. They also hear differently. Crickets and bush crickets have ears on the front, on their front legs, sort of near the, so just below kind of what would be the elbow joint, in us. You might think that's a strange location for a ear, and we'll get into what, the advantages of that organization in a moment. But just remember that that they're then, hearing off of, in fact, 2 tympana per year, right? And so they have these, these ears which have not 1 eardrum, but 2 each located on the forelegs. Whereas grasshoppers and locusts have a tympanum located on either side of the abdomen. Right, so very different position. We think different, developmental origins for these 2 kinds of ears as well. Among the cricket and bush cricket sort of subgroup of orthopterans, with, in this case, grasshoppers and locusts as an outgroup, there have been various attempts to sort of reconstruct the phylogeny of this group, and especially with respect to acoustic characteristics, because the question is, between crickets and bush crickets, which are 2 acoustic sound producing lineages within the cricket, among crickets, these are 2 groups that produce sound and hear sound. But a large number of crickets and bush crickets are also, do not produce sound and do not hear sound. So here you can actually look at a phylogeny by Gwynn et al. 95, which has subsequently come under. I mean, there've been more phylogenies that make different claims, and the 2 broad claims essentially around this are as follows. One posits, as this one does, that the ancestral trait is an absence of sound production and sound reception, and that you have 2 independent, emergences of sound production and sound reception. One in crickets, who produce low frequency calls, pure tone calls, and they seem to do this by rubbing the right wing over the left wing. Whereas among bush crickets, you, instead of pure frequency, a pure tone, low frequency sounds like a whistle is an example of a pure tone, low frequency call. Bush crickets will have high frequency broadband sounds like that first sound I played you, right? So, krr, 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 right? Many frequencies, broadband, and they tend to rub the left wing over the right. So the question then among crickets is, was this an ancestral trait with multiple lineages losing both sound production and sound reception, or with these small differences in how these 2 groups produce traits, do they represent multiple origins of hearing and sound and sound production? In the case of sound production, it happens through a file on one wing and a plucker on the other. So that goes krr, krr, krr. Now that you can kind of hear that in the call that I played you earlier, but with crickets and bush crickets, which produce these sort of whistle-like sounds, we think that striking actually sets portions of the wing in motion, and these then vibrate like a cymbal, like a cymbal as in the one that's used in an orchestra or a drum, to produce a pure tone resonant song. Okay, and this is a nice, beautiful close-up picture of the file and the plucker on the wings of a bush cricket. One of the specific animals that I work on is this absolutely beautiful, species called Onomachus, and I I'll probably have to rush through what we know about this animal because of lack of time, but I want to give you a quick glimpse about what makes the different model systems that we work on really cool. This is a bush cricket, right? And as if you've lost track, that's fine. But bush crickets are the ones, remember, that produce broad band high frequency calls. This is an unusual bush cricket because it produces low frequency pure tone calls. It sounds like a cricket. And so it's a nice system in which to look at whether at the evolution of song. Those are the ears and the front legs. You can see a close-up photo that we've taken and a CT scan on top as well. And one of the main problems that we study in our two field areas, one of which is in Meghalaya and the other of which is in the Western Ghats, is what's called the cocktail party problem, right? The question of how you recognize your call amid noise, right? How do you recognize your species call when you're living in a forest that consists of multiple species, some with calls like this, and others with calls like our Mycopoda, the first bush cricket I played. You can kind of hear that the first call is being masked by the second call, right? And there's a third caller now who's also being masked. So one of the questions that we look at is if you're listening to not just 4 calls, which I've just played for you, which are actually in a tropical rainforest, and you're hearing multiple representatives of all these species all calling together, it's actually deafeningly loud. And so we're interested in song recognition under those conditions of noise, but also in ways in which that acoustic noise may shape song evolution. One of the hypotheses around why Whistler has such a low frequency call is that, as a bush cricket, these live in, ground crickets live on the ground, true to their names. Bush crickets, slightly departed from their names, often live, at bushes and higher, right? So trees, these animals are found quite high up in the canopy on jackfruit trees exclusively. They live, poop, mate, everything on jackfruit leaves, as far as we can tell. And so one of the questions that we ask is, is this low frequency call something that effectively avoids too much overlap in the acoustic space with other bush cricket calls, and therefore does that provide selective advantage for unusual calls to evolve, especially in a tropical rainforest where you really have intense acoustic competition. And this is that low frequency pure tone call. It's absolutely beautiful. It's also, every sort of direction in evolutionary space also then has to battle other evolutionary constraints. So we look at other evolutionary constraints that also come with producing these low frequency pure tone calls. For example, these are hard calls. When you have a low frequency call, that's a high wavelength call. And the wavelength of the incoming sound is comparable to the distance between the 2 ears, meaning that the ears may not be getting a lot of very different information from one another about the potential location of a call, right? So you can tell which direction sound is coming from because sound either reaches one ear before the other, and some neural circuits compute that difference in timing or in the phase of the wave that reaches the 2 ears, and the other is just loudness, right? The sound that's here will sound louder in this ear than that. And if you're tiny compared to the wavelength of incoming sound, the differences between these measurable parameters between your 2 ears may not be strong enough to cleanly discern between these. So we do various different kinds of experiments. I'll briefly just skip over these so that you can see what we do with understanding morphology of the calling system. I also want to show you the behavioral work that we do, because that's perhaps the most compelling. This is an example of a female responding to the male call, and as you can see, she's kind of doing this whole body kind of jerk, vibratory jerk, in response to the male call. And she'll keep doing this for a while, for a couple of minutes. And so usually we find that female listeners just move towards the source of the call. We don't find that to be the case with these animals. It provides a very, very nice system then by which one can gauge the strength of interest of the female in the male call. Of course, Why they do this was for a long time unclear to us. It turns out that this whole body vibrational jerk, initially we thought it might relate to courtship, but the females will do this even if no males are present, as long as they hear the call. So we checked whether this tremulation sends out a vibratory signal. Sure enough, it does. We measured and assessed that using a laser Doppler vibrometer, which I'm happy to explain how it works if somebody has a question about this. But then we can quantify her tremulation, her moving of the branch, by using a laser that assesses the shift in frequency, or in Doppler frequency, of the reflected call, to get a sense of whether the surface it's directed at is moving away from the laser or not. And we can use sensitive laser vibrometers to even watch eardrums move. And this one was used to just quantify the strength of her vibratory signal, which, as you can see here, she responds with this vibration really out of turn with every chirp of the male call. But the strength of the vibration varies from chirp to chirp. And this has given us a really nice system on which we've now done further experiments, where we look at quantifying how much she continues to be interested in the call if you play frequency variants of the kind that you might see, upon which you might see selection acting in the wild. We look at temporal pattern variants to see how sensitive the call recognition is to temporal pattern variants, to get a sense of the strength of selection. So for example, we've done this work on now looking at the behavioral tuning of the call. That is to say, the call peaks at about 3.2 kilohertz. And the question is, how much does she respond to a 3.2 kilohertz versus, say, a 3 kilohertz or a 2.5 kilohertz song? And we see this nice broadband tuning curve in terms of how strongly she responds to frequency altered calls. When we're looking at a system where the calling song has evolved from high frequencies to low frequencies, you're looking at an interesting system where selection is acting at multiple levels. It's acting simultaneously on a song producing system and on a song receiving system. The song producing system is responsible for the calling frequency. But if the female is tuned to and listening to and responding to only a different frequency of call, then mutations that produce sounds away from that, from the peak of female spectral tuning will show reduced female responses. And so so this tuning curve then helps us understand how much leeway there is in mutational space for mutations in that affect call frequency, that is called the frequency of call produced, the extent to which it can compete or be attractive to females who are ideally tuned or show maximum tuning to another call, to another frequency of call. Okay, I'm going to skip ahead a little bit. As I told you, we also look at how the eardrums are tuned, because we're interested in looking at the animal as a whole. So we understand now that she's behaviorally spectrally tuned around the frequency of the call, of the male call. But when we look at her eardrums, remember I told you these animals have 2 eardrums. It turns out that the 2 eardrums in most crickets and bush crickets, one eardrum is functional and the other one's just a dud, or doesn't seem to respond to sound. But in this animal, you have differential tuning of the 2 eardrums per year. So you have 2 eardrums per year, and 1 eardrum seems to be specifically not a band pass filter like the behavior, but a low pass filter. That is, the eardrum vibrates to low frequency sound up to the animal's calling song of 3.2 kilohertz. And then the sensitivity of the eardrum just drops. If you play higher frequency sounds, this animal's eardrum is not even taking the input, at least the anterior tympanal membrane is not taking the input at all. It's not even conveying it to the hearing system. But there is a posterior tympanal membrane that does respond to higher frequency calls. So we're interested in looking at then how the segregation of calls of the hearing space into low frequency and high frequency bands sort of impacts the processing of hearing. But very interestingly, this low pass filter is so sharp. There's a sharp drop at 3.2 kilohertz. And it turns out that the drop coincides with with sort of where other forms of other sort of acoustic competition comes in from other crickets and bush crickets calling in the forest. All right. So I'm going to skip ahead, but just to say, therefore, that to give you a flavor of what we do in the lab, we have behavioral tuning, We have mechanical tuning, which is low pass. And we still have to, with neural work, fill in how it is that the lower end of the bandpass filter that we see in behavior is acquired by the system, right? But therefore, we do structural work, behavioral work, and ecological work. Okay, we measure neural responses in a variety of ways. In this case, we're hooking the nerve that comes in from the ear to what's called the prothoracic ganglion, a net of nerves that are processing the signal, and that send then an ascending nerve to the brain. And we're looking at then responses of these animals to 3.2 kilohertz. We see much stronger responses at high frequencies, 30 kilohertz, than to the animal's own call. And we think that that relates to the animal listening. The largest cell in the ascending nerve is what's called a T cell in most bush crickets, and that seems to be tuned to listening to predatory bats. Okay, so I'm not going to go into a lot of detail about why it is that we think tremulation may have evolved, the ways in which competition between or at least eavesdropping by bats off male calls produces then it essentially creates a cost for callers. And so we see all kinds of interesting responses, evolutionary responses to that. We see what are called satellite strategies among the males, where many animals don't call, and they eavesdrop on the female's vibratory response to the calling male to try and find her. We think that the female tremulating in the first place is a means by which 100% of females who fly towards males get eaten by predatory bats. But the calling, even though it's costly, is not as costly as her movement. And he's also smaller than her and less detectable. So we think that this intense predatory pressure upon females shifts the burden of movement onto the males in response to her vibratory signal. Okay. So what I'm specifically looking for is students who are interested in working on the neural leg of this story, and not just this story, other stories as well. We're interested in people who are doing electrophysiology, on this system. I'm going to skip ahead to just introduce one other system that we worked on, and then I'll close there. The one other system that we work on is the animal that I showed you in the first slide, Mycopoda. And the reason that we're interested in it is because the first call I showed you was fairly simple, right? It was cra, cra, cra. But we have, we see several different variants of this call, identical looking animals, but where males produce one of 5 different call types in the wild. And we're interested in looking at how these different call types emerge, as in what are the neural circuits that produce these different call types, what are the different pattern generators that produce these different call types. These have been looked at in some interesting degrees of detail in work done by Shonik et al. In the cricket system, but not in the bush cricket system. I'm happy to go into that in some detail again for anyone who's interested in understanding how that circuit works. Okay, so our longer term questions are, what are the selective forces shaping the evolution of neural systems? Is the coevolution between song production and song reception based solely on selection pressures, or are there also genetic links between these two systems, or neural links? Is there a corollary discharge from the song production system that's used in song recognition, and so on and so forth? How does song drive sympatric speciation? These are some of the questions we're specifically interested in Mycopoda. And we're also interested more generally in how songs are recognized, right? So how are songs are basically repeating patterns of sound in time, right? So if you have a krr, krr, krr song, how is that considered or recognized as a song, right? That's a question that we have. And therefore, we're also interested generally in how timing is assessed by neural systems. And we've started to get interested in questions of timing and quantitative and perception in other model systems as well, including zebrafish. So these are my acknowledgements, multiple grants from the DST, the CRG, Inspire, and Kothari fellowships have been entirely DST funded ever since I've returned to India. Ashoka, where I work, is provides an annual research grant, which is very, very helpful. And I have several collaborators, especially my postdoc mentor, Rohini, who started work on the behavioral ecology of the cricket system. And really, it's just a beautiful system on which to then try and look at the neural basis of the production of this behavior. And as I said, we also now work on zebrafish. We have a lot of people who are interested in working on our zebrafish system. I'm not going into it in much detail because I'm right now still looking for somebody, for specifically students to apply to our lab to work on the cricket system. And these are all the students in our lab. We now have six sort of graduate students, and multiple undergraduates who work in the lab as well. So I'll just close there and open up for questions. Thank you.